So, but before we go there, I like to just talk about the new approach methodologies a little bit. We hear this buzzword everywhere now on social media and podcasts. Would you like to just expand on the term a little bit so people can understand a little bit more what it is?
Yeah, absolutely. So you are correct. New approach methodologies everywhere, NAMS. There's actually been multiple publications on what NAMS is because some people use different terminology. But essentially, what it is any tool that can be used to generate human relevant data in place of an animal. So this can be anything from in silico. We have incredibly powerful AI models that can mine an incredible amount of data. Yeah, they can start to give us insights into safer, more effective drugs. And then after you have the data, you do need to prove that it works biologically. And this is where a subset of NAMs, which is called microphysiological systems, comes into play. So a lot of people think of organ on a chip organoids as NAMs. They're a part of NAMS, but it's a lot more than that. Where I spent my time was the organ on a chip and organoid space. But as I'm a part of the validation and qualification network, some of these bigger organizations, I'm able to branch out and see, how NAMS really encompasses so much more than I originally thought.
For people who are not in the scientific or pharmaceutical industry, the first impression they had about NAMs is like this is a way we try to avoid abusing animals or killing cute furry animals for testing. But what are some of the major reasons that we need NAMs and kind of shift away from animal models?
You know, I think that is important, right? Even as a scientist, no one likes to test on animals, but unfortunately, there just have not been better tools. And what the FDA is set up to do is make sure drugs don't get into humans that are unsafe and cause problems. And again, historically, we've not had tools other than animals to do that. With the advances in tissue engineering and bioengineering, we now have those tools to solve the biggest problem. 89% of drugs that are developed before clinical trials that are tested, in the lab, tested in animals, finally get FDA approval to go into humans, 89% of drugs fail. So I don't really know a lot of other places where you have a 10% success rate and keep going, right? But that's just been the way it is. And neuro, where I've spent most of my time, it's actually a 94% failure rate. So that's an even more difficult space that just shows you a mouse brain is not a human brain. And we now have tools that are more human relevant to be able to solve that. And as we do that, we're not going to replace animals tomorrow or next year. So I think it is important to remember that they are still a part of it. What we're able to do is start to replace and run aside animals. So you have to do less animal testing. And really, as that grows, as we prove that these technologies really are superior, I think that's when you'll see this replacement beginning to happen.
Like you said, human brain is obviously different from rat brain, but across the board, 90% failure, almost 90% failure is quite amazing that people still stuck with it for a long period of time. Where do you see, I mean, just looking at the failure statistics, where do you see the main reason animal models fail other than the obvious that we're not exactly the same,
I mean,
typically?
most often it's they fail to catch a drug that becomes toxic in humans. So you'll put this into animals, increasingly from mice to rats, then beyond that. And everything says, oh, this drug is not causing any problems. It's okay. It's not hurting the liver. It's not causing any kind of, heart cardiovascular problems. But then when you put it into humans, there have been a lot of examples where all of a sudden it's causing a big, big problem. And there have been instances where people have died because they missed something. Often people have really bad side effects, and it's just because they didn't catch something. There was such a disconnect in the biology. No, there was no way of predicting what was going to happen.